Any questions? And then oh, go ahead.
I was saying for the extra credit limit.
Yes.
Do we show up and write a lab report?
You show up, you'll write a lab report in lab. It will not be a lab report after lab sort of thing. So, and I'll tell you what the lab is when you get there. It's a surprise.
Okay. Wait, when is that?
There's the very last lab. So we have lab next week. We do not have labs Thanksgiving week. And then we have 1 week after that. That is an extra credit lab. It applies towards your lab grade. And it's a surprise if you decide you want to do it.
Is it individual?
No, this one's in teams.
And it can't hurt your grade, right?
What was that? It can't hurt your grade. No, of course not. Okay, questions? That would be a new form of extra credit. All right, well, let's go. We were talking about vitamins. We talked about vitamin A, right, for vision. I got that right at the end. We were rushed for time. I didn't really get to do a good job of my story. I'm not going to reattempt it. I'll look forward to next year, I'm telling you, and leaving more time. Then we also talked about vitamin D, right? And D is important for what? Signaling bone growth, right. All right, vitamin E is an antioxidant, so sometimes you'll hear people say that this is helpful against cancer. I don't really know off the top of my head. I would look for a meta-analysis on this, right? I would look at Cochrane and see if they have anything about vitamin E. But it is an antioxidant. And what an antioxidant is your body just doing metabolism, doing regular metabolism. Your body's going to make hydroxide radicals. Okay, so actually let me do the full Lewis dot structure here. So we've got a spare electron here on the oxygen that's really reactive. And where this is a problem and where it gets related to cancer is if you have some of these around and they're right next to your DNA, what they do is they go up to your DNA. I'm not going to show you the reactions because they're complicated. I don't even know them. But they go to the DNA and they grab an electron. Okay, and now the DNA is reactive and doesn't have an electron, and all sorts of things happen that cause bases to break down, you end up with mutations. You have repair systems in your body, but if you're hitting your DNA with a lot of damage, eventually one of your repair systems fails, right? So cancer is a disease of the DNA. Okay, it's of the DNA, at least mostly. I'm doing, with my research, I'm starting to get into doing a lot of reading about epigenetics. Does anybody know what epigenetics is? Some biologists tell me what epigenetics is.
How your genes affect you over generations, like different stress.
That is not it, but that's okay. Yeah. Yeah, non-genetic inheritable factors. So we have things like if you think about your cells, right? You start off as one cell, right? And you end up growing up into a ball of cells, and they're all the same. And at some point, they have to become different things, right? Those cells could become anything. At some point, they need to become muscle cells. They need to become brain cells. They need to become other cells, right? All various different cells that you have. And how do they do that? They have all the same DNA. So what epigenetics is about more than just this. But at one point, those cells differentiate, and they turn on certain genes, and all their daughter cells that they make, if it's going to make your liver, all those daughter cells express the genes to be a liver. And they're inheriting not different DNA, but how the DNA is turned on and off. Okay? So a lot of interesting factors there. And there are certainly, there are mutations to histones. So histones are what your DNA is wrapped around, right? There are mutations to histones that are drivers, actually, of cancer, which is really interesting. So that would be something that's the mutation is actually inheritable, right? But there's also how histones are modified, we'll talk about this next semester, affects what genes are expressed, all sorts of things like that. So it's not clear to me that I can completely say that cancer is only a disease of DNA anymore. And I think it's not really clear in the field yet, but I could see something happening there. Anyway, as you age, let's go back to the DNA. So as you age, has anybody ever heard something about cancer as you age? You've ever heard if you live long enough, if everybody lived forever, everybody would eventually get cancer. It's because you don't have the mutations that are causing cancer, but over your life, eventually you would, if you lived long enough, eventually you would acquire enough mutations that that would happen. Right. So it's bad to have something next to your DNA that's going to cause damage, because if you have enough of this random damage, eventually you're going to get something that is going to lead to cancer. So vitamin E is good as an antioxidant, and what it does is it can come in, it can grab that hydroxyl radical. Or it can grab that, actually it's going to sacrifice something for that. So if we think about, if we look at this aromatic ring part of vitamin E, okay, what do we know about hydroxyls next to aromatic rings? I haven't really talked about it too much. And we may not.
An electron withdrawing group?
It might be. I've forgotten. I should. It's been too many years since I taught organic chemistry, which is electron withdrawing. But you probably got a feel for things. You can do all sorts of weird things right here, because this is part of the aromatic system, sort of, right? You can stick some pi bonds up for that oxygen. I can stick up my pi bonds up here, and that can participate. And so it turns out that if you have a hydroxyl, if you have a regular water, hydroxyl on water, the pKa, if we talk about, well, let's not do water. Let's go ahead. Let's just do ethanol, right? So there's ethanol, right? The pKa of this is approximately 15. Okay, so not a very good acid at all. The pKa of this is actually about 10. Okay, so much lower, because it's stabilized with the negative charge because of the aromatic ring. All right, that's a negative charge, but we're talking about radicals. Radicals are also stabilized by having the aromatic ring next to it. And so what can happen here is I've got this going on, right? I can have this electron here come over here, right? And I can have this electron here come here. And what results from that reaction is a stable radical. So now I'm going to have one of these things. Okay, plus I have now neutralized my hydroxyl radical. I made it into a hydroxide. There's not all that much of it, so the fact that you have some base around it isn't a big deal. But this is a radical here, and this is a stable radical. Okay, so the way these work as antioxidants then is if you have something that is an oxidant, this is what we consider this to be an oxidant, okay? It's produced, but this is just produced in regular metabolism. You take certain drugs, you're exposed to certain things, you can have more of these. But this can be neutralized by vitamin E to make a new radical, but this radical is not reactive. We're just going to absorb it. That makes sense? See how it's an antioxidant? It's interesting. I don't know if I'll have time to talk about it. In second semester, when you think about photosynthesis, the start of photosynthesis, you need to get some electrons, and you pull them off of water, and they're tyrosines that are involved. Tyrosines are involved in pulling electrons off of water one at a time, and so the biology is taking advantage of the same thing here, right? That tyrosine here. Tyrosine has this structure, and so it can form a stable radical. And so at photosynthesis, right at the beginning, you have this happening. Okay. Yes?
Can you explain what an antioxidant is?
An antioxidant is something that gets rid of oxidants, but when we talk about oxidants, really, we're concerned with these radicals.
Okay.
They're just made by metabolism.
So about like stabilizing.
We're not stabilizing them, we're getting rid of them. We're getting rid of these reactive radicals, and for this one, in this case, we're replacing it with a stable radical for vitamin E. Okay. In other cases, you maybe just get, there may be no radical left afterwards. So, not second semester. We'll talk about something second semester that's related to that as well. When we're talking about glucose metabolism. Yes.
Where does the H go?
Where does the H go? Oh, that's a good point. This probably becomes water, doesn't it? Yeah, let's just make it water. Do they care really if you had H as a superior inorganic?
Yeah.
I remember asking that question once in class. It's oh, so where did that H come from? Oh, it's out in solution somewhere. He was my research advisor, and he was a lot of fun. Anyway, so let's move on. If you're okay, let's move on and talk about vitamin K. So vitamin K is important in blood clotting. You may have heard about this. If you haven't heard about it yet, when you have kids, when they're born, you'll be asked whether what things to give them. Maybe I don't know whether they have hospitals. They just give them all the shots, or whether they ask. Our kids were born at home, so we got asked, so vitamin K is one of the things. You don't have it. If you don't have enough vitamin K, you can have a bleeding disorder. Okay? Some small percentage of infants actually don't make vitamin K, and so they will just regularly, they'll just give every infant a vitamin K shot just in case, right after they're born. Okay, so we agreed to that, but so you give a vitamin K shot for that. And what is important for this is I've mentioned this before in the class. Vitamin K is important in the process of, remember we have the blood clotting cascade, that's all serine proteases. Okay, so vitamin K is important in the modification of those. So it's a post-translational modification. So I'm going to show you the process in a second, but important in the, what do we want to say? Carboxylation? I don't know if that's a real thing.
Sound good.
I've got a PhD in chemistry, so I can make up words. So it's important the carboxylation of glutamate residues on some of the serine proteases in the blood clotting cascade. All right, and so the process we're talking about here is you have, I'm going to show you this right here. So vitamin K gets used and then it gets regenerated. Can you guys see this okay? Is it too small? It can't be low, it's plugged in. Try harder. I'm going to turn down my screen just a little bit. Oh, that's the wrong button. Just a second. All right, I don't know. This may get a little dark in the video, but I'm trying to save the battery here since I'm having issues. So if we look at this, can you guys sort of see this? So look at, you see there's a cycle, okay? What I'm going to do is now I'll make it really big. So we've got a cycle going on. I'll make this bigger so we can better see the details. All right, so we've got vitamin K, which is a cofactor for enzymes, or a coenzyme. And what vitamin K is involved in here is we're gonna take it down, and so it's getting activated, or you can help activate an enzyme. Let's ignore warfarin for the moment. So, and then you have these glutamic acids that vitamin K is used, right? So we've taken this, we've got vitamin K down here, we've got the glutamic acid, and we're going to make this funny double carboxylic acid on the end. All right, so you've got your normal glutamic acid, it's got one carboxylate on the chain, right? And then you add another one. And for whatever reason, I don't know what it is, this is an active serine protease. So this is the active form. And this is our inactive form. All right, so we need vitamin K to do that. We've got vitamin K. You reduce the vitamin K, right? Well, let me point it out rather. We've got vitamin K. Part of this cycle is we take that vitamin K and then we reduce it, okay? Using it looks like it's NADH or NADPH we're going to use to do that. So we've reduced it there. And then that reduced vitamin K is used in the process of adding these, okay, adding these extra carboxylic acids to the end of the glutamates. And then our vitamin K is now in a new form that can't be used to reduce glutamic acids, right? It's not in the same form anymore. It's also not it hasn't been regenerated. So we have to regenerate it. And here what we have is we have a thiol. This might be glutathione that's being used. I'm not sure. But then we're going to regenerate right here our vitamin K. All right, so you've got a certain amount of vitamin K around. It gets used, it gets changed, and it needs to be regenerated. All right, when we think about, so we think about serine proteases. So we go back to vitamin K. So that's where vitamin K is important. And so people have taken advantage of this in terms of blood thinners. So here we have warfarin, which you may have heard of. It's a Coumadin blood thinner. And so we've got warfarin here, and we've got warfarin here. And essentially what warfarin is doing is it's inhibiting the regeneration of vitamin K. Okay, so you inhibit the regeneration of vitamin K, and then things can't clot as well. You don't clot as well because your serine proteases, not all your serine proteases are active enough. Does that make sense? How that works? Okay, so that's how the Coumadin-based blood thinners work. The Coumadin-based blood thinners are a little more of a, they're more of a pain than some of the current blood thinners, and people still get Coumadins. But what you have to do is, since it's all based on vitamin K, this is kind of a slow process, right? It's not an instantaneous. I take warfarin right now, I'm not going to have if I was taking too much warfarin, right? I wouldn't have a problem for a while because I'd still have my vitamin K that could run this process, right? But then it couldn't be regenerated after it was used up. Make sense? So an interesting thing is warfarin at low doses is used as a blood thinner, and at high doses it's used as rat poison. So you get those little orange cubes. I don't know if you've ever put them out or seen them. Don't eat them, and don't let your pets near them. Because essentially what it does is it's just a high dose of warfarin, and the point is that mice or rats will eat that, and then they essentially bleed to death internally because they can't clot. Questions? Okay, so if you're interested, well, here's the structure of warfarin. Okay, so it's not that huge a molecule. And if you're interested, I'll bring it down here by my blood clotting cascade. It's not all the serine proteases that get inhibited, it's just specific ones. So this has got a lot going on it. I have to look these up. Sorry. So what we're going to do is we're going to get warfarin inhibits 9A. So this one, 7A. Thrombin 2A. We'll do that one while I look for 7A. 10A. And you guys see 7A is right there. 7A is right there. So there's a lot going on here, right? But warfarin's going to come in and you're going to make this less active, this one less active, this one less active, and this one less active. Now, I know Xarelto. I don't know the other ones. My dad takes Xarelto. I don't know if it's an E. Xarelto is a relatively new, at this point, it's probably at least out 15 years. Xarelto is a drug, and what it does for blood thinning, and it actually just inhibits XA or 10A, okay? And it inhibits 10A in the kind of way that we are used to, we've learned, in that it's a competitive inhibitor. So you could take your Xarelto, and then you could stop taking your Xarelto and regenerate more quickly, right? Because you're just waiting for the Xarelto to clear your system, so your blood clotting 10A could be good again. Does that make sense? Okay. Questions? Vitamin K, blood clotting. You should know what warfarin's doing as well. Okay, just briefly, you don't need to draw the structures of the glutamates, but it's inhibiting the ability of vitamin K to be regenerated, right? And then vitamin K is needed to carboxylate those serine proteases so they're active. Yes, you don't need to know which ones. Warfarin, I don't actually know what kind of inhibitor warfarin is, but it's not actually inhibiting, any of these blood clotting factors directly. Yeah, I mean, so what's going on here is we have an oxidation. This is getting oxidized, right? We've got this disulfide getting oxidized in both cases. That would be interesting to look into because there's definitely, in terms of antioxidants, the antioxidant system I'll talk about next semester is glutathione, and it starts out looking like this, and it reacts with oxidants to become this, and then the oxidants are reacted not around. But I don't know if this is glutathione or not. That'd be my best guess. Okay, other questions? All right. Oh, then we're ready to go on and talk about membranes. Or membrane lipids. So I can see it a little better. All right, so we've got our membranes. So when you've talked about membranes before, this is kind of the basic diagram people first give about membranes, right? We've got polar heads and two nonpolar tails. We're obviously going to talk about this in more detail. We're actually going to talk about the functionality we get for these tails, what we get for the polar heads. But when we picture membranes, I hope you'll get to picturing them. If you're not already picturing them, you picture them something more like this. There's a lot going on in membranes. And so we do still have our polar heads with our two polar tails, but we're also going to have things like cholesterol in there. We have proteins that'll stick through. We have proteins that are associated with the surface. This is just non-covalently. We have proteins that are attached to the surface, like they're covalently attached to a lipid. All sorts of stuff. We can have, for cells talking to other cells, so this would be the outside, you have these oligosaccharides, so various sugar chains, more complicated than even what we're showing here, because they're all shown as like hexagonal sugars. But more complicated things. What else do you notice about like here versus here? We think about the outside versus the inside. There's more what?
Protein.
There's more proteins on the inside. I don't know if that's the case or not, but they're different is really what I want to get at here. The outside is different from the inside. Lots and lots of complicated things going on in cells and with cell membranes. And when you think about a cell interacting with the outside world, it's going to interact via the membrane. All right, so the compositions are really important. So we have a big pile of different membrane lipids that get used. And so we're going to start off with this diagram. We're going to talk about them as we go. What I want to do first, though, is I want to cross some things off this diagram. So the book I grabbed this for wasn't doing just membranes for this. We already talked about triacylglycerol, so that's not really what we're going to worry about for membranes. We're also not going to, they're super cool, but we're not going to talk about much. Okay, I guess I'll talk about them for a second because they're super cool. But these are archaeal bacterial lipids, so they're lipid membranes. What do you know about archaea? They're extreme environments, or at least the ones we've mostly found, right? I think it's been found that they're really kind of everywhere, various archaea, but we found a lot of these in extreme environments, and look at what they do with their membranes. Instead of having two tails that are you've got a membrane where you got two tails and you've got two tails and they're interacting with each other like this, but there's no linkage. They're covalently linked together. This is both leaflets of that membrane are covalently linked together to make it stronger. Okay, and you could imagine if you're living at 100 degrees or pretty close to 100 degrees Celsius, maybe you'd want something like that. But having said that, I will sadly cross that one out. We're not going to spend. They're cool, but we're not going to spend a bunch of time on them because they're not very common, at least in the terms of the things that we study. So what I want you to look at here is that we have a couple different classes. We've got kind of two classes of lipids, and then we've got two classes inside those classes. And we could actually break these down. We could take these classes and put them up here and put these classes down here, if that makes sense. So, but we'll go and follow this diagram. So we've got phospholipids and we've got glycolipids. Okay, and then inside the phospholipids, we have glycerophospholipids and sphingolipids. Okay, and I may say sphingospholipids. All right, there's some slightly different names out there, and I like to use the names that tell you all the pieces. So if we look at these glycerolipids, they both here and here, they have a glycerol and then two fatty acids. And then whether we're here or here, our polar head group, which is right here, is either going to be, contain a phosphate, or it's going to contain a sugar. Oops, let's do this one. Okay, that's where we have the difference between glycero and sphingo. Sphingo, or from sphingosine, actually is interesting. It's this big molecule. We'll look at it in non-box form. But it's the glycerol and it's one of the fatty acid tails all together. Okay, so we have for our sphingolipids, we've got that, and then we'll have a fatty acid, and then whether we're either talking about a phosphate for our polar group or we're talking about a sugar for our polar group. Okay, so that's essentially we've got four. You could imagine doing like I'm trying to remember for genetics. What is it? Punnett square. You could do a Punnett square, right? Where you've got phosphate and sugar, right? And you've got glycerol and sphingo, okay? And you could mix and match all possible combinations. And nature does that. So let's talk about the first one. And once again, so this is a glycerophospholipid. And sometimes I call them phosphoglycerolipids, just because I'm not, the literature is kind of a little loose with the designations there. But the general structure here is that we have two fatty acid tails. Okay, we have a glycerol base. That we're attaching things to. Okay, and then we have a phosphate in the polar group, in the polar head. All right, so those three things. All right, and so when you think about this, you've seen me assemble these things from glycerol, right? Glycerol looks like this. I could draw it like that. Sometimes I draw it more how it would actually exist in solution, and then I can combine that with fatty acids. I don't have a lot of room here, but what I could do is I could take that, right? I could take that or the water. I'm not sure which oxygen goes, but I could take that water off to attach that fatty acid to my glycerol, right? So I can do that here, and you can imagine doing that with a phosphate as well. It's glycerol, and you're sticking things on it. And so for glycerophospholipids, it's the glycerol, it has a phosphate involved in the polar head group, and then fill the other stuff with fatty acids. So where I'm headed here is, I don't know if we'll get to where I have an example quiz at the end, but I'm not going to expect you to draw these from scratch, but what I'll do is I'll give you all the parts. So I'll say, go ahead and draw me a glycerophospholipid, and there'll be a picture of glycerol on the page, and there'll be some fatty acids on the page, and there'll be a sphingosine on the page, and there'll be some sugars on the page. And so based on the name of it, you have to connect them up correctly. Okay, get the correct parts together, but also do the correct bonding. All right, so this is where we have our normal esters. Questions? Okay, so in terms of looking at these sorts of lipids, here are some examples of things that can go into that polar head group. So when we draw that polar head group as a little circle, right, with the two tails coming off of it, this is what this is the polar head group is. This flips over this way, away from the tails. And that's what you get. And so with that polar head group, you could have the phosphate and just a hydrogen, right? And if you were to draw one of these from scratch or something, I don't care that you get a polar head group correct or that you know all these. I just want to show you some examples, okay? If you're asked to draw this, just put the hydrogen on because it's the easiest thing, right? But there's various things that happen depending on what this does. You guys have heard of choline before, right? Sometimes it's a serine, sometimes it's a sugar with phosphates on it, okay, which would be a confusing one because the next thing is going to be sugars for the polar group. So that's a mix of things. But there are all sorts of various groups that can hang on after that phosphate as part of our polar group. Notice, though, that they are all polar, right? The worst of these is maybe, well, it depends on what these R's are. We've got some more, right? Here we have another phosphate, that's pretty good. And then depending on what these R groups are, that might be tough making it polar. Questions?
How is H polar?
H isn't polar, but you got it right next to a phosphate. So when I look at that overall, the phosphate just dominates everything if it's just an H. If I add something huge onto it, it doesn't dominate so much anymore. Oh. Before we talk about that, of course, you now have the hint. Let's talk about what our 5 most abundant elements are in biology, right? 5 most abundant elements in biological systems. All right, what are they? Right. So we have hydrogen, we have the big 4 from organic, right? Oxygen, carbon, nitrogen, okay? And then we have phosphorus, okay? And we see phosphorus a lot. We just saw it in our membranes, right? We have that membrane. That's a membrane you find around the membrane lipid. The glycerophospholipids are lipids you find around kind of any animal cell, all right? However, phosphorus is not super abundant. It's in fact not very super abundant in the universe. So this comes from, I like to listen to this guy named Isaac Arthur, who does a lot of science and futurism, and he talks about things like the Fermi paradox. Anybody know what the Fermi paradox is? What's the Fermi paradox, Chloe? Oh, well, take Jacob.
Given the abundance of planets in the universe, the absence of life.
Right. Why aren't there any aliens? Okay, there's so many planets in the universe that there's got to be a huge that regardless of if life is rare or not, there's so many planets. Why aren't there aliens out there? Odds were first are really low. Okay, and so one of the solutions for that, they have a bunch of solutions, right? People come from all sorts of reasons, but one of the things is phosphorus is really rare. Okay, and in the universe, and that we happen to have a fair amount of phosphorus on Earth. And so maybe we got lucky in the way that be it for the solar system, the way the Earth formed, that we got enough phosphorus for life to evolve here, and that lots of other planets that could have had life don't have phosphorus. Okay, there's a ton of other solutions as well. Okay, so phosphorus is important, but it's rare, and it's rare on Earth too. It's hard to get in some places. All right, so let's say you're a plant, right? You're an animal and you can't get enough phosphorus. You can go move over someplace else and eat some phosphorus over there, right? Okay, if there's not, but you're a plant and there's not enough phosphorus in the soil. What do? Die, tough it out in some way. So plants have evolved to use less phosphorus, okay? Plants have evolved to use less phosphorus because it's rare. All right, and they can't move to some place where there's more phosphorus or go grab something, eat something that has phosphorus in it. At least most plants can't. All right, and so what they have in their chloroplasts, they use sugars as the polar group instead of phosphorus. Or I should say monosaccharides. All right, so this is an adaptation to what environment most plants find themselves in. And so this is going to be an example. This is a specific example of one. You don't need to know the specific example. Okay, but what we would call these is, here, I'm going to make sure I'm matching my terminology with our figure up above. Where do they put galactolipids? No, I'm not going to match their terminology. Galactoglycerolipids, okay? All right, I'm going to use a terminology that's going to tell you what the pieces are in the name. So galactoglycerolipid. So it has a glycerol as the base. It has a galacto, hopefully you recognize that as a sugar thing. It's from galactose. Okay, and in fact, yeah, that's galactose. Okay, so this is an example of that. Questions? So this is our polar head group. All right, and so, I don't have examples of other things you can hang off of that. But like what we had up above, this isn't the only thing, right? You could have a sugar, you could have a collection of sugars, you could hang other things off of that as well. But if I give you this an example on quiz, just pick the simplest one, okay? All right, so now we're going to look at our different base for these molecules. So we can, instead of glycerol, we can have a sphingosine as the base. And I say the base because it's what you're attaching things to. Sphing. And it can be the base for membrane lipids. And so we'll talk about the specific ones in a second, but what I want to show you here is, first off, this is the structure of sphingosine. So you see that it has a fatty acid tail, a fatty acid sort of tail. It's not a fatty acid, right? But it's got this long hydrophobic tail, one of them already built into its structure. You need to recognize the structure and be able to tack things onto it. You do not need to be able to memorize that sphingosine. I won't give you sphingosine and then something with 10 carbons here that's not sphingosine. Okay, that makes sense. So the other fatty acid, look how it's attached. Look where it's attached. How is it attached that's different from what we have with glycerol, glycerol lipids? Those other ones have esters, and this is it's a peptide bond. It's an amide, right? Okay, amide, peptide bond, I don't care which. And then on the other, down here on the end, and notice that the phosphate groups, the polar groups are always, not always phosphates, the polar groups are always on the end, okay? For this, obviously, there's a specific end. Don't put them in the middle, okay? But if you draw the glycerol and you put it on the top one versus the bottom one, they're not different, so it doesn't matter. Questions? Yes?
So if there's sphingolipids under like glycolipids and then phospholipids, is the thing that differs the like phosphate group versus like having a sugar group?
Here we're going to have two of these. We're going to have phosphate versus sugar.
Okay.
But I could have grouped these, I could have just as well grouped these as glycero and sphingo and then broken them out for categories. So I'm presenting these in a certain order and they're presenting the diagram in a certain way. But I talked about a Punnett square, but I waved my hands in there and I didn't draw it at all. So we could think about glycerol, and we could think about sphingosine, and we can think about phosphate, right, attached, and we can think about sugar. And there's not a hierarchy here in terms of those classes in the way that I'm presenting it in a certain way. It's in the figure in a certain way.
Okay.
But what's going on is you get all of these combinations. Did that address your question?
Mm-hmm. So how I guess would we know if you say like sphingolipid, like how would we know whether it has like a phos?
It'll say phosphosphingolipid, or so this is just sphingosine itself.
Okay.
We're going to go take a look at examples, but it'll be phosphosphingolipid or sphingophospholipid, whichever way it is. Okay, so here we have some different examples of things that you can hang off of a sphingosine. All right, and so here we haven't actually presented, we're not in a category of phospho or not phospho or galactose so far. But so here we've got, this would be a phosphosphingosine, phosphosphingolipid, phosphosphingosine, and then this would be a galacto. And so the difference here is, well, I'll give you two quick examples. Oh, I don't want to do that one first. Maybe I do. Okay, I want to do this one first. Let's start. We're going to talk about sphingomyelin. Does anybody know what sphingomyelin is important for? Nerves, right? Okay. So sphingomyelin. This is a sphingophospholipid. So sphingophospholipid. Okay, sphingofospholipids are important, that they're structural in nature. Okay, sphingomyelin, which is really the only example we're going to talk about, sphingomyelin wraps around the axon for nerves, and it helps to insulate it. What was that?
Is it the myelin sheath?
It's the part of the myelin sheath, yes. All right, because I have 2 minutes, I'm going to just say that. But all I want is, it's a sphingophospholipid. You could put it together from parts, okay, and you can tell me it's structural. That's what I want. And then going up here, so sphingogalaktolipids, let's actually see what name they used up here, and I'll match that. Unless they call them sphingolipids. Nope, I'm gonna not use that because I want the names in it. Okay, so we're going to talk about sphingogalaktolipids are involved in signaling. Okay, and so here the difference is we don't have I don't have a full structure here, but this is an example of your blood groups actually, not blood clotting groups, or they cause clotting. So when we think about your blood groups, you're either A, B, O, right? In terms of that. And what those are is on the outsides of your red blood cells, what you have is you have either this set of sugars, right? If you're O, you have this set of sugars in that shape. If you're A, you have the same set, but then you have an N-acetylgalactose on the end of it. And if you're the B, your blood type is B, you have this O shape plus galactose on it. All right, so that's the difference between those. So you may have heard, you've got, sometimes you hear people being AB, right? AB blood type would be you'd have some of these and some of these. Okay. I'll probably talk about that in more detail on Monday. You guys have a good weekend. If you have any questions,